Hello, everyone. My name's Brett. I'm a second year grad student in Dr. Rule's group. And so I'll give a brief research update um, into what I've been working on. And so my research lies under Objective 3.1a of the PDT focal area, which is to develop experimental strategies to determine novel sequences for diagnostic and therapeutic proteins. And so under that, to design a build, test, learn cycle and cell-free expression for proteins. And so generally what my work has been focused on is experimental strategies to rapidly express and characterize nanobody proteins for protein engineering workflows or protein screening. And so, like I said, nanobodies are the proteins I've been working with mainly. And so nanobodies are single domain antibodies, which are derived from heavy chain only antibodies found in camels. And so in this figure on the right, you can see the top figure. This is what a traditional IgG antibody looks like, where you have both your heavy chain and light chain domains. And then the bottom graphs depict these heavy chain only antibodies, which can be found in camels. And the variable domain of these antibodies is what we refer to as an antibody. And so nanobodies, similar to traditional antibodies, they have very high levels of affinity and specificity towards their target. And then their small size gives them numerous other favorable characteristics and applications. And so again, my research is focused on experimental strategies to rapidly express and characterize these nanobodies. And so for rapid expression, we're using cell-free expression. And so as the name implies, cell-free expression is a method of protein production that allows us to generate proteins without the need for live cells. And so when we generate, when I create these reactions, there's three components I'm mixing together to generate our protein. The first part is the extract, which is harvested from cells, and this is what contains all the necessary machinery for protein production. And the second part is the supplemental mixture that just contains all of the other substrates and cofactors needed for protein production. And then the third component is the DNA, which of course contains the code for which encodes the protein of interest that we're interested in. And so the big advantage of cell-free expression over traditional cell-based expression methods is that it can be fast and easily scalable for a large number of unique protein sequences. And so that's why it's useful for the screening phase in producing proteins. Okay, and so the next part I want to talk about is, so once we express our protein and generate our protein, we want to characterize its affinity. And so to characterize affinity for our nanobodies, we're using biolayer interferometry, or BLI. And so BLI works by, we have these little biosensors, and these biosensors can record or can measure the interference pattern of white light off of the end of the surface of the biosensor. And so as proteins bind to the end of the biosensor, it causes a shift in that interference pattern of light, which can be recorded as a signal over time. And so this allows you to monitor biomolecular interactions in real time. And so when we do that, we can generate a sensorgram that looks something like this on the bottom here. And once we have that data, we can fit it to kinetic models and determine our KD value, which is going to be our value that we associate with the nanobody affinity. And so one important advantage that's useful with BLI is that it can work with unpurified protein samples. And so this means that I can use cell-free to express my protein rapidly overnight, and then I can measure its affinity without purifying the protein next day. Okay. And so putting everything together, this is what my workflow looks like, my complete screening workflow. And so I first use Golden Gate assembly to assemble a linear expression template. And so Golden Gate assembly allows me to introduce mutations easily and cheaply to generate, generate the templates for whatever proteins I want to test. And then I can put that into cell-free expression to generate my nanobody. Once I have my nanobody, I can measure its concentration using a fluorescent tag directly from the cell-free reaction. And then to characterize it, I first take my BLI biosensor and I immobilize the antigen onto the end of the biosensor, and then I can equilibrate that biosensor in blank lysate for a few hours. And then once I have that, you can dip the biosensor back into solution with your nanobody to measure the association, and then put it back into blank lysate to measure the dissociation. And then from those curves, you can generate or calculate your K on and K off, and then your affinity parameter being the KD value. Okay, and so putting all that together, I first wanted to kind of verify the accuracy of this screening tool. And so the nanobody I've been working with is what we're referring to as the I3 nanobody, which has been provided to me. The sequence has been provided to me from our collaborators at the University of Iowa. And so, the graph here on the bottom right shows an example sensorgram of the data I collected with this nanobody in BLI. And so I did this, I tested this using my screening protocol and then compared it to the data they had also provided me with. And so the graphs here on the left show the comparisons between the K on and K off values and the KD value that I calculated versus our collaborators. And so you can see here, there's some discrepancies between the K on, K off, but the overall KD value, I calculated to be very similar to what they had provided me at 2 micromolar. And so after that, I moved on to using the screening tool to test a variety of different nanobody mutants. And so with this I3 nanobody we've been working with, it has a very low affinity at 2 micromolar. And so ideally, we'd like to engineer it to have a higher affinity. And so they had also, so our collaborators had also provided me with 12 different nanobody mutants to test for improved affinity. And so I was able to use my screening tool to basically rapidly express and characterize these 12 mutants. And from this initial screen, we can see mutant 10 had a slight improvement in affinity at 0.75 micromolar, but overall not a dramatic increase. But okay, and so the main limitation in the screening protocol that I've developed so far has been the low cell-free expression yield. And so ideally when doing these BLI assays, to get good quality data, ideally you want to include analyte concentrations that are above and below the KD value for that interaction you're working with. And so if we're getting relatively low cell-free yields compared to the KD value, it can be, that limits us so that we're not able to test those higher concentrations. And I can see this in the data I've collected, and this is what this graph looks like, or graph shows on the right here, and that as I exclude higher concentrations from the kinetic fit, the error associated goes up significantly. And so, so because of this, one thing I've been working on is trying to improve the yield of my cell-free expression. And so one thing I've done that provided me with the greatest increase in my yield was just doing codon optimization. And so the way I tested this was I just took my nanobody sequence that I'd been working with and ran it through IDT codon optimization tool several times. And then I measured its expression in cell-free. And you can see from the original, I was able to nearly double my expression just through that codon optimization, which was interesting. Okay, and so for my future work, I want to continue to improve my cell-free yield. And then I also want to do another test where I use the same ligand-loaded biosensor for multiple runs. And so instead of using a different biosensor for every analyte concentration, I use the same biosensor for several different analyte concentrations. And that way the amount of antigen or ligand protein that's bound to the end of the biosensor will remain the same for sample to sample. And then I also am looking for ways to improve throughput through automation using liquid handlers and stuff like that. And then in the future, I also want to explore different characterization techniques, such as carbon nanotube probes being developed in our lab, and then also alpha LISA. So, okay, and with that, I'd be happy to try and answer any of your guys' questions. Thanks.
Thank you, Brett. Questions? I'll start with one. So why camels?
There's I think it's just that is the organism that they've been found in, that those heavy chain only antibodies have been found in.
It's the only animal that has those?
Well, I think it's the family Camelidae, or I don't think it's just camels, but like within that family of and then there's been some kind of similar single domain antibodies even found in like sharks and stuff like that too. They're different than antibodies, but there's been some other interesting ones too. So
very interesting. Mark, you're muted.
Thank you. Yep. Hi, Brett. Nice presentation. I'm just wondering how the antigen is attached to the tip of the probe, and if that sort of binding chemistry might block the antigenic site on the monobody, the nanobody.
Right. So with the system I'm working with, the antigen is purchased and it's biotinylated. So we use streptavidin-coated biosensors to bind the antigen onto the protein. And so there's a variety of different biosensors that you can use to do that chemistry. You can make your own, there's lots of different functionalized biosensors you can just purchase, like the streptavidin ones that we use. But then there's also you can just use there's ways you can make your own like custom biosensors. But yeah, like I think it is part of assay development where it's something you sort of just have to test and keep in mind that, like, the orientation of your protein could make a difference, and if your protein is binding to the biosensor at a weird orientation, that could affect your affinity measurements. But
okay, thank you.
Zinji?
Hi, Brad. Very nice presentation. I wonder, like, how many fragments do you normally assemble together as you use a Golden Gate?
Yeah, good question. The uh, for the testing I've been doing, I've just been using 3 fragments. Has worked for introducing the mutations that I need.
Mm.
I know Ryan, who's also in our lab, has been doing testing to try and see how many fragments you can actually do total. I've just done 3 just because that's what's been useful or what's been needed for me. But yeah, ideally you could assemble up to 10 or more. But
Yeah.
yeah.
I'm curious because you mentioned you want to just introduce a single point mutation. That's what I understanding.
Yeah.
And in that case, why not just use the simple like a PCR and the overlap extension? Because whether this is a easier approach to introduce just a single point mutation.
Um,
Mm.
yeah, I guess that could work. I guess the idea is if we have several mutation locations and we want to test different combinations
Mm.
easily, we could just assemble those with, if you have the fragments for each of those, you could assemble it easily that way.
Because that's why I'm asking if you assemble multiple fragment, I totally understand. But if it's just a single point, I thought maybe PCR by using set-directed primer will be easier.
Yeah, I think like going forward, the Golden Gate will be useful if we're trying to increase the number of mutations and test different combinations. But yeah, for what I'm doing right now, I probably could do it that way just as easy.
But do you worry about the contamination with the wild type protein?
Um, yeah, I mean, I've thought about that. I mean, so far I've just done like simple like Sanger sequencing to kind of verify that I'm generating the template correctly, and it seems to not be an issue. But yeah.
Okay, other questions for Brett? Any other questions, comments?
I don't have a question. I don't have a question, but I can highlight that this is an example of a project of something we want to do here at EPSCoR more, which is this cross-institution, cross-research lab. So this is in collaboration with the WADUS group at University of Iowa. So they developed this nanobody long ago with a collaborator, aren't really keen on how well it binds, have kind of done in silico design of where it could be better. And then this project came along and Brett was able to generate those candidates, test their affinity. And now they've come back with a long list of other things they want tested. And so now it's trying to figure out what's the rate limiting step and how we can do more of these and design more. And then Ratul is going to work on the computational side. So it really is kind of a confluence of groups, and it's exciting. We want more of these types of interactions.